You know, for thousands of years, humans have been partnering with life to create things we need and love. Think bread, cheese, wine. But what if we could go beyond just using life and actually start rewriting it? Well, that's exactly what we're going to explore. We're diving into the incredible world of biotechnology and the powerful tools that let us engineer life itself.
So, here's our game plan. First, we'll nail down what biotechnology actually is. Then, we're going to open up the genetic engineering toolkit and see how all the magic happens. We'll walk through how scientists put these pieces together, check out some mind-blowing real-world applications, and finally, talk about the huge power and responsibility that comes with all of this.
All right, so first things first. What exactly is biotechnology? Well, at its core, it's a partnership between us and the microbial world. And it's a partnership that's gone from an ancient art form to some seriously precise modern science.
The official definition is actually super broad. For literally thousands of years, we've been using microbes to make everything from food to medicine, all without really knowing what was going on at the genetic level. That was classic biotech. But then, in the 1970s and 80s, everything changed. A revolution started.
And that revolution was recombinant DNA technology, or rDNA. This is the absolute game changer. We're not just using cells anymore, we're getting in there and editing their instruction manual. We can now literally take a gene from one organism, like a human, and pop it into a totally different one, like a bacterium, to get it to make something useful for us.
This diagram right here, this is basically our roadmap for the whole process. Over on the left, you see we start with a vector, which is usually a little circle of DNA from a bacteria, and then we have the DNA that contains the gene we're interested in. We cut and paste them together to make what's called recombinant DNA. Then we get that new DNA into a host cell, which starts dividing and making a clone army. From there, you can either make tons of copies of the gene itself, or get those cells to produce a protein for you. We'll be coming back to this blueprint, so keep it in mind.
Okay, so to pull off these amazing feats, scientists need a very specific set of tools. I want you to think of it like a geneticist's toolkit, one that's designed not for wood or metal, but for the molecule of life itself.
So, let's look inside this toolbox. We find three absolutely essential items: a pair of molecular scissors to cut DNA with incredible precision, some delivery trucks to carry genes into cells, and a genetic photocopier that can make billions of copies of a gene from almost nothing. First up, the genetic scissors. These are actually proteins called restriction enzymes. Bacteria originally made them to defend against viruses, and they are amazingly precise. They basically scan along a strand of DNA and only make a cut when they find a very specific sequence of letters. This precision is what makes all of genetic engineering possible.
And check out how they cut. You can see here it's not a clean snip straight through. The enzyme makes a staggered cut, leaving these little single-stranded overhangs. We call them sticky ends. And because these ends are complementary, a piece of DNA from, say, a human can easily pair up with a piece from a bacteria, as long as they were both cut with the same enzyme. It's like having perfectly matched puzzle pieces that just want to snap together.
Next in the toolbox, we've got our delivery trucks. These are vectors. Okay, so you've cut out your gene of interest. Now what? How do you get it inside a host cell? Well, you splice it into a vector. The most common kind is a plasmid, that small circular piece of DNA you find in bacteria. It's the perfect vehicle to carry a foreign gene into a cell and make sure it gets copied every single time that cell divides.
And finally, the genetic photocopier, the polymerase chain reaction, better known as PCR. Just imagine you're a forensic scientist with just a single speck of DNA from a crime scene. That's the kind of challenge PCR solves. It's this revolutionary technique that takes that one tiny sample of DNA and makes billions, literally billions, of identical copies in just a couple of hours. It's the engine behind so much of modern biology.
The way PCR works is through a cycle of 3 simple steps, repeated over and over. First, you crank up the heat to separate the DNA strands. That's denaturation. Second, you cool it down a bit so these tiny DNA fragments called primers can bind to the ends of your target sequence. And third, a special heat-proof enzyme called DNA polymerase gets to work, building new complementary strands. Each cycle doubles the amount of DNA, so you go from 2 to 4, 4 to 8, 8 to 16. It's exponential growth.
Okay, so our toolbox is ready. We have our scissors, our delivery trucks, and our photocopier. Now for the really cool part. Let's see how scientists actually use these tools to build a brand new genetically modified cell from scratch.
So the first major hurdle is actually getting our engineered vector, our plasmid with its new gene, past the cell's natural defenses. Cells have membranes and walls, and they don't just let any old DNA wander in. So scientists had to get pretty clever. One really fascinating trick is called protoplast fusion. As you can see here, scientists use enzymes to gently dissolve the tough cell walls. What's left behind is called a protoplast. It's basically a naked cell. And once they're in that state, you can get them to fuse together, letting them mix and match their genetic material to create totally new hybrid cells. Now for challenge number 2, finding the needle in the haystack. The truth is, the process of getting DNA into cells is really inefficient. You might end up with millions and millions of cells, but only a tiny handful have actually taken up your gene of interest. So the big question is, how on earth do you find that 1 in a million cell?
This is where a brilliant trick called blue-white screening comes into play. Just look at this petri dish. Our goal is to find the white colonies. Here's how it works. The plasmid we use as our vector has a gene that normally turns the bacterial colony blue. But, and this is the clever part, we insert our new gene right into the middle of that blue gene, which breaks it. So, if a cell successfully takes up our engineered plasmid, it can't make the blue color anymore. It forms a white colony instead. The blue ones are failures. They got the original plasmid, but not our new gene. It's just a fantastic visual way to spot the winners.
So, we've gone through all the intricate steps of how you build a modified cell. But the real magic is in the why. This isn't just a fascinating lab trick. It's a technology that has completely reshaped our world in ways you might not even realize.
The impact on medicine alone has been just staggering. Before rDNA, insulin for diabetics had to be extracted from pigs and cows. It was expensive, and it could cause allergic reactions. Today, we have bacteria working as tiny, living factories that produce pure human insulin. The same goes for human growth hormone, and for creating safer, more effective vaccines like the one for hepatitis B, which is made by yeast cells carrying a tiny piece of the virus.
And agriculture has been just as transformed. By inserting a gene from a bacterium, we've created crops like Bt cotton that produce their own natural insecticide. That means farmers can use way fewer chemical sprays. Other plants have been engineered to resist herbicides, or, like the very first GMO tomato, to have a longer shelf life by just turning off the enzyme that makes them go soft.
Of course, a technology this powerful doesn't come without some really big questions. The ability to literally rewrite the code of life forces us to face some complex safety issues and pretty profound ethical dilemmas.
Sure, there are strict safety rules to keep modified microbes from getting out of the lab, but we also have to think about the bigger picture. Could these pest-resistant crops accidentally harm beneficial insects, like bees? Could these new genes escape into wild populations? And on the ethical side, who should have access to your personal genetic information? And how do we make sure these amazing new therapies are accessible to everyone, not just the wealthy? Look, recombinant DNA technology has given us an almost unbelievable power to shape life itself. The benefits, from life-saving medicines to more sustainable crops, are absolutely undeniable. But as we move forward, the most important work might not be happening in a lab coat. It might be in the conversations we all have, together, about how to use this incredible power wisely, ethically, and for the good of everyone. What principles do you think should guide us?